Right, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh, and a very good day, everybody. So, welcome back to SID3019. So today, what we're gonna do is gonna continue with our lecture. Okay, but probably I'll just do it as quick as possible, because as you might have noticed that my voice is actually a bit weird. So I just recovered from a flu and a cough, of course in a fever over the weekend, but thankfully it's not COVID. So we're just gonna, but because my throat is still a bit sore, so I'm just gonna go as fast as possible, but of course it will be as thorough as possible as well. Okay. So now, as I have mentioned last time, that we will have at least on plan 7 synchronous lectures and then 7 asynchronous lectures. Okay, so all learning, so including mixed lecture, tutorial discussion, and lectures and tests. Okay. And on week 7 last week we already did our quiz. Okay, quiz number 1, and on week 14 we will also have another quiz. Alright, and because quiz 1 covered lecture 1 to 5, quiz number 2 will cover lecture number 8, which is today's lecture, until week 13. Okay, so week 13 will include hopefully from an NPRA. So I'm inviting an NPRA expert to actually give a lecture related to this topic. Okay, and then finally on your final exam, during final exam week, so meaning that we'll complete all 14 weeks, then you will have one study week, and then you will enter your final exam weeks. Okay, so we will have your final exam then. And of course, today is the 26th of April. We will have another 2 more asynchronous lecture, and then we will have your mid-semester break. Okay, it's a bit late, so it's in the middle of May, and it will be concurrent with, if I'm not mistaken, during the Eid celebration. Okay, and then after which we will continue 1 synchronous lecture, and then 2 synchronous lecture by invitation. Okay, at least that's the plan. I've yet to get any confirmation from these 2 parties, but I will try my best to actually get them to contribute or to share their knowledge in this particular section. Now, this is also as what I have requested you guys to complete this survey. Okay, so please, it will be very helpful for me and for my colleagues. So we are actually looking at different ways of teaching. So one way is to have a live lecture with all the live questionnaires, okay, or live feedback questionnaires. The second one is, as per this recording, we will have embedded quizzes and some activities that for you to do during while you are listening to the lecture. Okay, so for live lecture, when you were to select the intervention, there will be intervention A, and for this recording lecture, it will be intervention B. So I will be very grateful if you guys can actually help me and participate in the survey. Now, so before we go into this lecture, okay, so just a very quick one, what we have covered so far. Okay, so we already have 5 weeks of lecture, and those 5 weeks of lecture involve a bit of basics of proteins and peptides, what are the structures, and the forces that are involved in holding the structures, different type of structure, alpha, beta, random coil, and so on and so forth. Okay, we also looked at very briefly on the two pathways where you can synthesize peptides and proteins. So one is a synthetic pathway, another one is a biosynthetic pathways. Okay, we also looked at immunity and vaccination, also in brief. It's by no means in very detail. We looked at classical vaccine components such as epitope, so based on peptides or proteins, and also adjuvants such as immunostimulants, carrier molecule, and so on and so forth. We also looked at antibodies. So once you have produced a vaccine candidate, you deliver the vaccine candidate into your subject, or animals or humans, and the body. What are the steps, the processes that are involved in producing antibodies in the body? Okay, and finally, we've looked at one application of an antibody, which is a rapid test kit, and it's for this specific examples. What we looked at is on COVID RTK. Okay, so the picture on this on the right is actually showing different types of vaccines that we are actually did not cover. Okay, so we have over here live attenuated. Okay, so of course we did not cover that. So we've covered protein subunit or peptides. Okay, we did not look at RNA per se, but I did show you guys an example of RNA vaccines by Pfizer. We did not look at anti-DNA vaccine, but a good example of a DNA vaccine is the dengue Vexia. Okay, Vexia, yeah, it's a vaccine against dengue virus. You can Google that and find the information on why, even though it's a good concept, but it's dengue Vexia is actually a subunit. Okay, the dengue virus based on DNA is not commercial yet. Okay, and traditionally we can also have inactivated virus. Okay, this is one of what COVID vaccine is. Okay, the COVID vaccine from Sinovac. Okay, so they are using inactivated virus, and we can also use a non-replicating viral vector. Okay, this is one of the example in COVID as well, and replicating viral vector. So these are some examples of vaccines that can be used, or different technologies of vaccine that can be produced to develop immunity for humans, and so on and so forth. Okay, so this is the lecture for week 8. Okay, so what we're going to focus on today is the identification of peptides and proteins, so to say, in terms of epitope and antibody. Alright, now so once we have all these vaccines, so we've looked at on how to actually synthesize this vaccine, right? So the next step is, once you have them, how do you actually analyze the synthetic proteins or peptides? So in this particular lecture, the main focus is just on peptide and protein vaccines. Okay, it will be a very long lecture to actually touch on how you analyze every single thing. So the primary focus here is just a peptides vaccine or a subunit protein vaccine. Okay, so if you notice, it's a bit jerking, so I actually paused before I cough. Alright, so going back to the lecture, when we're talking about how do you actually analyze peptide and protein, so a scenario that you need to consider is something like this. Okay, so say for example you have designed a peptide. Okay, peptide based vaccine for whatever diseases. Okay, of course you need to recall peptide can be an epitope of a vaccine, so that's why we design peptide. Okay, because you're going to use it as part of a vaccine component. But in order for the vaccine to be as effective, you will need to use, or you need to add an adjuvant, right? So also, of course, it's already been covered. Now something that you need to ponder is these two questions. Okay, of course you can ponder more than what these two questions, but this is just an example on what is the basic thing that you need to think about. if you are going to develop a vaccine. So one, how much peptide or adjuvant should you use? Okay, so here we are talking about the ratio. One is you need to think about the amount, the quantity of the epitope that you need to put in. Okay, so that's what, because there's a threshold pretty much. So even if it's a complete vaccine, it's a threshold. Say for example, if a vaccine construct, okay, including the adjuvant, you need to deliver say 50 micrograms, for example, per dose, then you need to know this value. But how do you get to know this value? How do you know that this value is actually the correct value? Okay, and then second one is, how do you ensure that your peptide has the correct sequence? So in trying to answer this question, okay, so I put it here in order to answer the question is, how much peptide adjuvant should you use? Okay, and it might be a very unscientific, but the truth is, I don't know, we don't know, nobody knows. Okay, why do I say that there is nobody knows? Is because of two reasons. Okay, so the first one, oops, sorry about that. Okay, so number one is because one is you are always not sure how efficient is the epitope in eliciting immune response. Okay, so how can I say this? Efficiency. Okay, efficiency of epitope. So we know that the epitope, once delivered into the body, will be processed. Okay, and so also in the lecture number 4, I've talked about antigen processing. Okay, so we know that the antigen or the peptide will definitely be processed, but we have no idea about the efficacy, the efficiency of the antigen itself. We know it works, okay, but towards the end of the day, once you develop a vaccine, you deliver into your body, what your body produces is not just a singular antibody. Okay, so even in one peptide epitope, the body might actually produces like, I don't know, 10 or 20 different antibodies just based on this one sequence. Okay, so that's why that's why I put it here. We don't know. So that is something that you want to investigate in the future. Okay, so as I mentioned, yes, we know that it can produce an antibody, but there are still a lot of unknowns in the field. For example, if say an epitope has 10 amino acids, would adding another 2 amino acids, so you already have 10 amino acids. Okay, and then you add another 2 amino acids, does it make a difference? Okay, so because towards the end of the day, it all depends on the antigen processing. So we know the processes, we know the steps of antigen processing, but we don't really know on how our body select whether this side of, say you have 12, remember, if this length is 12 amino acid, whether the body processes the first 6 amino acids, or the last 6 amino acids, or probably the middle bit, or probably, you know, if you have 12 amino acids, if you move in 6 amino acid sequence, then you will have at least 12 different antibodies. That is if the antibody detects 6 amino acids. Imagine if the antibody actually detects 8 amino acids or even 4 amino acids. So these are a lot of variations, and even in the literature, they will always give you a range of length of the epitope that can be used as an antibody. Okay, for the development of antibody. So there's a lot of variation. So that's why I said here, you don't know. We will never know. At least for now, we don't know. In the future, maybe someone will discover about it, but so far, this is something which is still unknown. So the key is to okay. So this is the lecture that I've talked about. You know, looking at antigen processing. Okay, so these are the processes. So we know the process, but we don't know how is the selectivity during the process. Now, so if you add or subtract number of amino acids, you might result in a different path. Therefore, we might produce a different antibody, and consecutively the effectiveness of the antibody will be different. Okay, and reason number 2 is we are now sure how effective is the we are not sure how effective is the combination of the two, meaning the adjuvant and the epitope. So some adjuvant are very good, but they are too extreme if you combine it with a different epitope. So a lot of variations, and when we are talking about adjuvant, there is no one single adjuvant. There are many types of adjuvants that can be used for vaccine development. Even the common ones that we are using, alum adjuvant. Okay, so when we are a kid, we had our BCG antibody, BCG vaccination. So in that vaccine, most likely alum adjuvant is being used. So this alum adjuvant is actually serendipitous. Okay, and this researcher just combined it and then suddenly it works. So there are a lot of research going on regarding with alum, whether it's alum crystals, whether it's different types of aluminium. Okay, but you can still use it. So because of these various degrees of unknowns, thus a reason to develop a preclinical studies to actually investigate the effectiveness of each component, meaning that some people during clinical studies, animals normally is being used, and we deliver a different quantity of the vaccine candidate to the animals to gauge the optimal level, because you don't want to inject too much vaccine, because again any disease, if you inject too much, will cause harmful effects. Similarly, if you drink too much water, okay, it will be harmful to your body. Okay, you can Google it up. I'm not telling you lie. If you drink too much water, sorry, if you drink too much water, you can also develop a disease. Okay, all right. However, if you are looking at question number 2, okay, something that you are pondering, how do you ensure that the peptide has the correct sequence? That is something that we can do, and that is the focus of this particular lecture. Alright, so over here I have listed down 3 options. Okay, 3 options by which you can actually look at whether the sequence of the peptide that you synthesize are correct or not. Okay, but for this lecture I've only hoping to cover 2 options, and on next week's lecture we will cover the third one. Okay, so let's move into the first option. So option number 1 is what we call as EDMAN degradation. Okay, you might have covered this during your second year in a different subject, but I'll just put it here again to actually reiterate on the ways by which you can use to identify the sequence of peptides. Now, this can be either if you synthesize it chemically in the lab, or even if you synthesize or you produce it using a biosynthetic approach. You can also use the same technique. Of course, there will be slight variations in the protocol, but towards the end of the day, this is the technique, or these are the techniques that currently being used to actually identify the sequence of peptides. And of course, it's not limited to analyzing epitopes. Okay, so in the future, if you're doing any peptides or proteins based research or discoveries, okay, and you want to understand on or identify what is the sequence of the peptides or protein, then you can use the same methods. Okay, so option number 1, EDMAN degradation. It's a 3-step process consisting of number 1, coupling of phenyl isothiocyanate, okay, or PITC, to the N terminal of the peptides or proteins. Okay, so that's the first step. The second step is to cleave off the last amino acid, so the N terminal amino acid. Of course, the PITC have already conjugated to the first amino acid. Now the second step is to the cleave off of this particular amino acid via cyclization in a strong perfluorinated acid such as trifluoroacetic acid. Okay, and then you will form this 2-adenylino 5-thioisolinone base structure. I will show you in the next slides on the structure itself. And number 3 is by converting this intermediary structure to a more stable structure by the formation of phenyl thiohydrodentoin. Phenyl thiohydrodentoin. Okay. Or PTH. So PTH can then be easily identified. It's very stable and is very easily identified using HPLC. So by just looking at, of course, you need to run a standard, and then you will know the retention time of that particular PTH amino acid. Then you can know. Okay. And EDMAN degradation is a very highly accurate technique as long as the N terminal is free. Okay, so of course you need to have a linear peptide, or even if it's not globular, it's fine as long as there is a free N terminal. Then you definitely can use EDMAN degradation to analyze the peptide. But we will see in the next slide. Okay, we will see later. So this is the chemical process of EDMAN degradation. So first off, we have this phenyl isothiocyanate. Okay, so that's a phenyl, what we call it isothiocyanate. Cyanate is normally like that. Okay, you have S there, so it's thio, so this one is just cyanate, that one is just thio. We call it isothiocyanate is because it's not a cyanate in the sense that a carbon triple bond to nitrogen, but the carbon are actually double bonded to the nitrogen, and then you have another double bond to the thio group. So phenyl isothiocyanate. Okay, of course, so you have your first reaction, a conjugation to the N-terminal amino acid over here. Okay, at a slightly alkaline pH. So you can have a conjugation like that, but in a very acidic, such as using TFA. Okay, you can form a cyclization into this structure to alanino 5-thioazinolinone. Okay, and then finally under aqueous acidic condition. So this one is more on neat. Okay, so this one is more on aqueous, whereby you will have water molecule reacting and reorganizing the thioazolinone structure into a phenyl thiohydrodentoin structure. Okay, and if you see here, this is the R group. So the R group over here corresponds to the to the R group of the amino acids. Okay, so for glycine, the R group is just H. For alanine, then you have a methyl group, and so on and so forth. Okay, and by doing this, say for example on this structure over here, you will also before you run your EDMAN degradation sample, okay, in this one, so you will definitely need to run your first PTH standard. So if you have 20 naturally occurring amino acid, then you need to have a 20 standard PTH standard corresponding to each amino acid. Okay, so say for example, as shown here, if your PTH standard has an retention time of roughly about 15, 17, 16, 17, 17 and a half minutes. Okay, So when you run your EDMAN degradation sample, you want to have a peak that has a similar retention time. Okay, what is shown here is actually if you do not get the same retention time, then you know that the first amino acid is definitely not the same as what your PTH standard is. Okay, so in order for say for example, if this one is a glycine PTH, then to know whether your first N-terminal amino acid is glycine is for you to have similar retention time. Okay, alright. Now so that's about it. A very simplistic information about option number 1, which is EDMAN degradation. Now moving to number 2, which is using HPLC-MS or MS-MS tandem mass spec spectrometry to actually identify the sequence of the peptides or proteins. Okay, so now this technique is best used for a purified sample. Okay, well, of course, if it's not purified, then you can always purify it first before actually run it. Okay, otherwise the process is more complex than is discussed here, because you need to do a lot of analysis, you need to use a lot of libraries, you need to use like a lot of libraries that might have been pre-prepared, or a specific library, for example, if based on the enzymes that you actually use to help in analyzing the peptides or protein sequence. So it's very detailed, so I'm not gonna go into that. So just gonna go like touch on very brief on the technique and how you actually read the technique. Okay, so MS-MS requires sample preparation. HPLC-MS also requires sample preparation. This is to make sure that the peptides or proteins that you get is actually purified, so it will be easily identified. Okay, you can still use a semi-purified samples. It's fine. There's no issue as long as it's partly purified, and normally it will be a successful a technique. Otherwise, it will be more complicated than what I'm going to show you now. Okay, so the step is very easy. The first step is to do a sample preparation. If it's not purified, then you need to purify it, and so on and so forth. Number 2, you need to run it. So you run on HPLC-MS or tandem mass spec system or OBTRAP. So there are a lot of systems, but what I'm going to show you is just HPLC-MS, the most basic system. And number 3 is to do a post-analysis to actually identify what is the sequence of peptides or proteins. Okay, so we'll go through one by one. The first one is sample preparation. So first off, to know how to actually prepare the samples, you need to first know or understand, or at least, you know, if you do it on your own, then definitely you know whether the sample is a synthetic sample, whether you synthesize it in the lab, or whether you are actually producing the peptides or protein using a biosynthetic pathways. Okay, using cells or cell-free assay, or even if you produce it in plants. Okay, so that's the first step. Next, of course, you need to have a certain degree of purification, and depending on whether you are doing a synthetic or biosynthetic approach, the process is different. A synthetic approach is very simple, because normally when you synthesize something, the purity is not as bad as if you are using a biosynthetic approach, because a synthetic approach is in a very controlled manner, so it can be easily purified using a chromatography. For example, you can either use a size exclusion chromatography, or you can use affinity chromatography. Very simple. For a biosynthetic pathway, you can either use precipitation method. Okay, meaning that when you have a peptides in a solution of cells, you can precipitate out the protein, thus kind of like semi-purifying it. You can also use chromatography, affinity chromatography, for example, using histidine nickel histidine tag, okay, tag system, or you can also use gel electrophoresis. Okay, so these are and of course you can always combine one of these methods, or there are more methods that you can actually use to purify proteins. You can always use them to actually get the highly purified or best purified proteins as possible. Okay, I'm talking a lot now. I have a lot of mucus. And then what do you do after that? So once you have it purified, for a biosynthetic approach is you need to digest them. Okay, so why do you need to digest them? Because biosynthetic protein normally do not have free N-terminal amino acid. Okay, in comparison, if you are doing a total synthesis, you always have a free N-terminal, provided that you don't do any post-synthetic modification, such as doing a cyclization or whatnot. So if you have a linear peptide, then it's very easy, it's very straightforward. But for biosynthetic, normally you don't have a free terminal due to post-translational modification. So this one depends on the organism that you use to actually synthesize the protein or peptide. So you might have a phosphorylation, you might have alkylation, you might have a glycosylation, and so on and so forth. So you need to remove all of these post-translational modification to actually be able to identify the protein or the first amino acid. Okay, otherwise what you can also do is, imagine if you have a protein like that, okay, with an N-terminal with a glycosylation, for example. Okay, so glycosylation. Okay, but what you can also do is using trypsin to cut the sequence randomly, and thus creating a starting point. Meaning that if you use a trypsin might cut here, cut here, cut here, cut here, and cut here, and here, and here, and here, and so on and so forth. So by cutting all these segments, of course, now once trypsin cuts in the middle of the amino acid, then you will have a free N-terminal amino acid. Okay, so you use and this technique is what we call as a shotgun technique, because when you have a shotgun, when you fire a shotgun, it will just randomly go anywhere. So it's not as if you are using a pistol. So pistol has a higher accuracy, shotgun has a less accuracy, so it goes everywhere. So that's why we call it as a shotgun technique. Now if you have something like this, then it will definitely be more complicated, because now you have not just one sequence of peptides. Now you have multiple sequence of peptides, and therefore you need to identify each sequence individually, or you can do it concurrently. So yep, but I'm not going to touch about the more complex ones. We'll just look at on how you actually use HPLC and mass spec to easily identify a simple sequence. Okay, so and finally, for synthetic ones, you can just straight away go to MSMS or HPLC-MS. But for biosynthetic, once you did your digestion, you can straight away proceed with the mass identification. But okay, by no means synthetic you cannot go through this digestion step. You can, especially if your synthetic is very long. For example, if you are synthesizing 100 or 200 amino acid peptides or proteins, you can always start off the digestion and then you go through to the mass analysis. Okay, let's look at how you actually analyze it. So once you have once you have digested the peptides or proteins into a smaller fraction, so what you will get is probably something like this. Okay, imagine that this is your full length of your protein. Okay, once you have added trypsin to digest this, you'll have one segment there, another 2, 3, and 4. Okay, so in this example you will have 4 different peptide sequences. Okay, and then what you do is, of course, you need to as best as you can to isolate this segment individually if you can, or otherwise you can always use tandem mass spec, okay, tandem mass spec or MSMS, to actually identify each component individually. Okay, so we will not go into MSMS in this lecture. We will see if we can actually go into the next lecture, or if we don't have enough time, then if you are interested in MSMS technology, you can always talk to me, or talk to Dr. Tay. So there are a lot of lecturers in UM who actually knows about this system. Okay, but what I want to show in this slide is basically, once you digest it, of course you will have segments of shorter peptide. Okay, and then even if you inject everything on mass spec, what you will see is something like this. Okay, so you will see the peptide will roughly have the same intensity. Okay, because imagine if you have one strand of peptides, you cut it into small 4 different fractions, you always have equal ratio or close ratio between all fractions. Okay, so that's why I'm pointing to this piece over here. Okay, but what if you only take one of these and then you identify in mass spec? This is what you will see. Okay, I will go into the next. I will explain about just one sequence after this. Okay, because the theoretical sequence is always known, meaning that if you synthesize it, so of course you know the sequence. Okay, the mass of the complete intact sequence can be predetermined. So meaning that before you digest, you just take a little bit of sample, you analyze it, then you will definitely know the parent mass of the whole sequence. Okay, so normal mass spec without digestion or fragmentation, and fragments can then be logically identified by mass tracing, or what we call as mass peptide mass fingerprinting. So what do I mean by that is this. Okay, if you look at this, so this can be an example on one peptide. Okay, so even if it's already digested, you take one shorter fragment and then you analyze it. What will you get? What will you see? This is what you will see. Okay, especially if you are doing MS, this is what you will see. You will see masses, some of them at a very high intensity, some is at relatively low intensity. So if you get something like this, how do you identify what is the sequence? Okay, so of course the first step is to identify the parent ion from undigested peptide. So this example over here, the parent peptide is about 1498 Dalton. Okay, so that's the mass. And then number 2, identify all peaks of interest, the daughter ions, okay, which normally has significant peak intensity. So in this case, you'll have 1, 2, 3, maybe 4, 5, 6. Okay, you have six peak mass of interest. And then the third one is to randomly start mass tracing and then identify the corresponding mass to mass of amino acid. Okay, how do you do that? Okay, so just focus on this. Okay, focus on this masses. Okay, and then I will move to the next slide. Okay, so these are the masses that I've circled just now. Okay, so these are the daughter ions that has the highest intensity. So what you do is, you know that this is the parent, that is the smallest, just based on your run. What you do then is you take the difference between these two masses. For example, if you do if you have 1079.335, you minus by 980.287, then you will get a mass difference of 99.048. Okay, Dalton. So which amino acid is this? Then you will look at here, list of amino acid and residual mass. Okay, residual mass or residual mass within AMU. and then find 99.04. 99, oh, this is the closest. Okay, 99.07, even though it's not accurate to the dot, but 99.0, which is the closest to 99.0 here. So you know that this particular sequence corresponds to a valine. Okay, so one of the amino acid. Similarly, if you take that mass different, then it will be a serine. Okay, if you take that mass different, if you take that one, if you take that one, if you take that one, then it doesn't mean that you will always get a mass corresponding to something. Okay, but in this case, that one is valine, that one is serine, that one, if you look at that, is about 120. Okay, but over here there is no 120, so this is not corresponding to any of the amino acid. Okay, if you go back here again, okay, so this one is nothing, so that is valine. Okay, so that's 99. That one is serine, can't remember what's the mass. Well, unfortunately this one has nothing. Okay, so there's no amino acid correspond to this section. However, there is an amino acid corresponding to this particular region. If I'm not mistaken, it's tryptophan. Okay, so even though the daughter ion like this one is big, but it might not correspond to anything, so you always need to go back and forth. And then of course, how do I know this one is correct and not that one? Is because you are doing a mass tracing. Okay, so you know that first amino acid is valine from C-terminus, and then you have serine, and then you have something. Okay, so this something can be either this or that, but of course after that you will look at the next one. So if this one has something, then this one is the correct daughter ion. If this one has something, then this one is actually the correct one. And by analyzing this, then I know that this is correct, and this one, if I'm not mistaken, is alanine or glycine, I can't remember, but there is no amino acid corresponding to this particular region over here. Okay, so that's how you know. So from there you can identify the sequence of amino acid. Okay, it's actually not that difficult. I will do a tutorial based on this. I will just give you an examples, and then you will work on it, and we will discuss if what you actually did are correct. Okay, and of course these two information you don't have to memorize. I will provide it for you for your final exam. Okay, so this will be part of your final exam thingy. And of course, that's it for today. The third option is to do NMR spectroscopy. For this one is actually very straightforward. The complexity comes on the NMR analysis, which we will cover next time. Okay, so that is all from me for today. It's about 43 minutes. Before we end, I need to try and reiterate that I'm hoping that you guys can contribute, and hoping to contribute to this survey. Okay, some references, and these are the list of references that I've used to build these lecture slides. Okay, that is all from me. Thank you, and have a good day.